our Nano in Business series, looking at public and private partnerships to help advance pre-competitive activities. I've got great pleasure in welcoming our three speakers this afternoon. So we have Joanne Shatkin from Vireo Associates, Daniel Hewson from Fiberlean, and Dimitar Prodanov from IMEC. So we'll make a start, and Joanne, if you're ready, we can begin with you, if that's okay. You should be able to share your screen.
Yes, Sean, hopefully you can see my cover slide here.
Indeed, perfectly, Joanne. So I will let you make a start, and thank you ever so much for agreeing to present today.
Oh, I'm thrilled to be here. As Sean said, I'm Joanne Shatkin. I'm president of Vireo Advisors, and we're a new member of the Nanotechnology Industries Association. And I'm thrilled to be here. So I'm kicking off this webinar to talk about how public-private partnerships are advancing pre-competitive nano safety. So I'm going to talk about a few such partnerships that we're working on. But first, if I can just take a moment to introduce ourselves. Vireo Advisors is headquartered in Boston, Massachusetts in the States. And so it's morning for us at the moment. But we've been working since 2013 to bring better technologies to the market, recognizing that that uncertainty about safety demonstration for new technologies got in the way of commercial acceptance. We've been working on ways to reduce the risks of innovation and address the environmental health and safety requirements, both market-driven and regulatory-driven, and connecting our partners to the resources they need. So, you know, with new technologies, with nanotech, it's not always straightforward to sort out how to demonstrate the safety. And so you can see in the list of organizations below, and these are just the public organizations that we've been working with, that we have both research institutes, academic organizations. We do have a lot of commercial partners. I did not include them here. But we are working to help navigate the environmental health and safety landscape and manage the safety aspects. But today, I'm more going to focus on the work that we do to build and work with consortia to gain market and regulatory acceptance for new technologies, novel, nanoscale, and also bio-based technologies. So we work at that intersection where technology meets policy, and it drives us into these public-private partnerships where it's a win-win-win for all of the participants. So industry needs the data. The investors in private foundations want to see their innovations get to market, as do companies and entrepreneurs. Government agencies have a need to see the data and want to help develop sound methods that they can rely on for safety demonstration. And then, of course, we need the researchers in academic and research institutes to help us generate the methods and the data. So everyone wins in that type of collaboration, including the cost sharing aspect. So we work internationally. We're in North America, but we're working with companies all over the globe on several continents, and they have interest in entering markets all over. So we do pay attention to what's emerging in different parts of the world, and have successfully been able to build and manage multi-organization projects, and develop biological testing strategies, and cutting edge testing, physical and chemical characterization, and also a focus on sustainability. So we've been involved in a number of consortia. The one I'm going to focus on today is the Alliance for Food Safety Acceptance of Fibrrillated and Crystalline Cellulose, or AFSAC. But later today, I'll be part of a workshop on this New Harvest Safety Alliance. So I did want to just mention it. You might have heard that the Singapore Food Agency just authorized the first cellular agriculture product. And so we're engaged there and trying to build a consortium to come up with new safety testing methods for that technology, which has nanotech applications as well. We're also working on a safer manufacturing toolbox. Hopefully I will have time to mention that. And then a couple of projects with the Organization for Economic Cooperation and Development, or OECD, Working Party on Manufactured Nanomaterials, where we've been collaborating with diverse organizations on advancing the science for safety demonstration by alternative test methods. So using alternative testing strategies and looking at adverse outcome pathways. So this is the example I want to talk about today is the Alliance for Food Safety Acceptance of Fibrillar and Crystalline Cellulose. And we're a consortium that is structured as a pre-commercial safety evaluation. It's a collaborative public-private project, both in the partners who join, who include governmental organizations, a private foundation, research institutes, academic organizations, and seven companies. And the driver behind the work is that these novel forms of cellulose materials are similar and are going to behave similarly in the gastrointestinal tract to conventional forms of cellulose. And so that because celluloses are generally recognized as safe by the US Food and Drug Administration, we can conduct side-by-side testing for these novel forms alongside the conventional forms and demonstrate the safety of the novel forms too. So we've been developing data sets and are doing this side-by-side conventional animal testing, which is what food regulatory agencies want to see, but also looking at, can we come up with some 21st century toxicity testing, new methods, novel methods that are cheaper, faster, easier, and be able to read across to those, and then also be able to leverage all the data that exists from, historically. So this is a multi-aspect project in that we are having to develop methods and also do conventional studies, good laboratory practice, animal studies in commercial certified laboratories, developing both data as the basis of safety and supporting data, and also being able to read across from conventional to fibrillated and crystalline forms, and then have methods for additional forms as well. So we've been implementing a testing strategy. We're now in the very last piece of that here. At the end of 2020, the ADME testing. Obviously, the methods development was the first thing we had to do, and then we were able to do the conventional testing. Now we're focused on these final set of tests, and just a quick schematic to show you the alternative strategy that we employed, where we had 6 partner materials and 3 study materials and ran all of them side by side in this simulated gastrointestinal digestion to do non-animal or in vitro toxicity testing, to have a number of different endpoints that are critical now to look at the next generation of forms. So not only demonstrate that these 9 forms are similar, but have now the basis for the next generation of materials that are in development. So we had 3 materials from the University of Maine, a hardwood cellulose nanocrystalline powder that we compared to a conventional food fiber additive that's been on the market for a long time. And then in our alternative testing, we were able to compare the industrially produced materials and to the forms in our animal tests that allowed us to read across. This way, we didn't have to engage in animal testing on the commercial forms. So I don't want to spend a lot of time on it, but only to say that of all the different tests that we ran, we saw no adverse effects in any of them for any of the forms that we tested. And we're able to demonstrate that even up to 4% of the diet, that both the cellulose nanocrystals and the fibrils behave similarly to conventional cellulose. And so we saw no concerns up to 4% of the diet. So we're now taking these data sets and bringing them to different regulatory agencies, including the Food and Drug Administration, to allow for their use both in food and as food contact materials. So this now forms the basis for the next set of work that we want to do, which is novel material safer manufacturing toolbox. So for as I mentioned, the next generation of materials is going to look a bit different in order to use many types of nanomaterials. They need to be functionalized in order to be mixed into composite materials. And so when we have these surface modifications or functionalizations, we don't want to go through a 4-year research project with 10 partners to do all this work. Again, what we want to do is come up with a more streamlined approach. And the idea is that we will select some targets for those functionalized materials, come up with some methods to look not only at food and food contact uses, but a variety of endpoints, including occupational exposure and other types of uses, other markets that the materials might go into. And so then we will have methods that we'll develop and work to standardize. So there will be a toolbox and a data set demonstrating the safety of these materials. Hopefully we'll have some structural alerts that will be helpful for innovators to be able to look at different types of functionalization for their materials. And we'll have those methods that will get out in the public domain. And I did want to share, in this project, we've been publishing the work that we did. The very bottom one, number 12, there is the roadmap that we started with, where we got a lot of input into what are the priorities, what are the gaps in knowledge, and we used that to build up to the most recent publication came out just this week, actually, demonstrating the safety of sulfated cellulose nanocrystals. Having these in the public domain increases trust levels for the market and for regulators, and so that's a key underpinning point. Just to reiterate, the value that we see in these public-private partnerships is that for new technologies, you need to ensure that the methods you're using are valid, and that's a role that governmental organizations and private foundations can help to play to get the industry into the market successfully and reliably. And companies need the data, but it's a lot when you have to develop the methods. So by collaborating in a pre-commercial way, everybody wins, and we generate these data and we put them out in the public domain to help get the whole industry toward commercialization. I did want to give a nod to my team, without whom I couldn't get all this work done. And I'll stop there, Sean. Thanks.
Okay, that's perfect. Thank you ever so much then, Joanne. So we'll move seamlessly then to Daniel Hewson from Fiberlean, who indeed has been one of the people working in one of the collaborations with Joanne, and he can sort of give you their story. So Daniel, you should be able to share your screen and yep, that's absolutely perfect.
Did that come up?
Yep, perfectly.
Okay, good. Okay, hello. So I'm Daniel Hewson and I work for Fiberlean Technologies. Like Sean said, I'm going to talk you through our regulatory journey and share some of the work that we've done and the results that we've had from it. But first I'll tell you a bit about Fiberlean. So Fiberlean Technologies was formed by the coming together of two of the world's largest mineral companies, Imerys and Domya. And this joint venture began in 2016 and has grown to a company of 66 employees. and is now one of the world's largest producers of microfibrillated cellulose. Our R&D facilities are based in Cornwall in the UK, but we also have offices in Belgium, Sweden, and the US. Fiberlean's success comes down to the cost-effective method used to produce this MFC mineral composite. The process uses a robust, scalable, vertical stirred wet mill, proven to work on an industrial scale. The mineral particles aid the process by acting as micro-sized grinding media, reducing the energy required to produce an equivalent degree of fibrillation without mineral. And this method allows us to produce 12,000 dry metric tons a year. We have further capacity currently under construction. We're also able to supply a no mineral product and products with either high or low solid contents. And our R&D efforts include partnerships with universities, research institutes, equipment suppliers, and various consultants and regulatory specialists, such as Vireo advisors and the NIA. We have established sales in paper and packaging, and have recently moved into the construction industry. This graphic here kind of highlights the potential applications for Fiberlean, especially in a time when industries are seeking green materials and sustainable alternatives to the finite and unfriendly. MFC is organic, it's biocompatible, and it's chemically versatile. Functionalization of the fibril surface allows additional control of desired properties and tunable dynamic behaviors. So this is what our current clearance status looks like. US Fiberlean has an existing substance under TSCA. We also have food contact notifications and an FDA GRAS application in progress as part of the Vireo-led consortium that Joanne just spoke about. We're also recognized in Canada, Fiberlean was accepted by the Health Canada for use in contact packaging. And in China, we have recently had an application approved by the NHC, also for food contact packaging. And then in Europe, we have recent approvals. So in Germany, from the BF, they approved the use of Fiberlean in food contact packaging under clearance 36 and 36.2. And the same application was submitted to the Dutch authorities, who have approved the Fiberlean under Chapter 2 of the Dutch Commodities Act, covering paper and board. And as I said, Vireo advisors and the NIA have supported this work and our clearance activities from the very beginning. So we're very grateful to them for that. This timeline gives you a rough idea of how long it's taken. I know the joint venture began in 2016, but the developmental work began much sooner than that, and regulatory work with it to ensure that the safety of those handling the materials were indeed safe to do so. And so we started with migration studies, literature reviews, and dust particle studies, and these all came out very satisfactory. And today, I'd like to talk about some of the characterization work and the latter, or the most recent migration study that we've used, and that have contributed to the equivalent clearances highlighted there at the bottom. So the characterization work that we did, the aim was to compare Fiberlean MFC to the MFC found in an existing food product. And these were the various techniques that we used to characterize both the physical and the chemical properties. And then these went into the reports. Yeah, I'm not going to have time to cover all of these, so I'll talk about a few of the highlighted ones and just go over some of the data. Just a bit of background to the SEM fibril counting work that we did. This was to give us size distributions of the materials, and the SEM images were collected at 4 different magnifications: 500, 2000, 5000, and 150,000 times, to provide subsequent suggestive quantitative data of fibril width distribution. 20 micrographs were examined at each magnification, and fibril thicknesses were measured manually using dedicated image analysis software. Duplication of counted fibrils and fibers was reduced by defining an acceptable size range for a given magnification. Measurements outside the defined range were ignored, and the size ranges for each magnification were based on the overlap between the varying magnifications. And then from this, the cumulative number distributions were derived for each sample by calculating the number of distributions for all the magnifications. And the number distributions were calculated by combining the normalized number of fibrils common to each lower magnification with the number common to the next consecutive higher magnification. So the natural, or the existing MFC that we used, we extracted from ketchup. Ketchup is a highly processed food, the homogenization of which liberates the fibrils that you can see in the images on the right. And we used a simple dilution and centrifugation technique to extract these fibrils. And then from the SEM work, we measured the fibril widths. Of course, you can tell from these images that you can't see the ends, so we could really measure the widths. And morphologically, the two types of MFC proved to be very similar, depending, of course, on the refinement used. Size profile can be obtained to match. And the ketchup MFC is the light blue line that kind of runs to the outside at the finer end and then through the middle at the coarser end. And here we have size distribution profiles generated by laser diffraction. And again, they show a very similar trend. And the ketchup sample again is the light blue, shown here against various Fiberlean products. And again, there's a very good agreement between the size distributions. Here we have Valmet fiber analyzer results. And these are the various length measurements made by the Valmet fiber analyzer, and again show a good agreement between the Fiberlean products that we've made and the ketchup MFC. Here we have the IR spectroscopy, and these results show strong chemical profile similarities, typical of cellulosic material. The peaks found at around 3,322, 2,884, 1,070 reciprocal centimeters are all associated with the oxygen-hydrogen, carbon-hydrogen, and carbon-oxygen bonds found in cellulose. Some of the peaks in the Fiberlean samples are masked by the stronger mineral response, which are the peaks found at 1,411 and 873 reciprocal centimeters. So overall, the characterization that we did showed strong similarities between the physical and chemical properties of Fiberlean MFC and MFC extracted from ketchup. So moving on now to the migration work that we did. This was our approach, and it consisted of preparing sheets based on a standard furnish that's listed there on the left. And then similar sheets were made containing MFC. And these sheets were then subjected to the migration tests according to the standards listed there, the EN 645 and the EN 647. And then for analysis, listed on the right, the BFR in Germany required that we use scanning electron microscopy as the characterization method. And as far as we were aware, there are no standard methods that exist for conducting a comparative analysis of samples using SEM. And there's also no automated identification sort of counting software that exists for MFC. And these were problems that we had to find solutions to. So we made the sheets with and without MFC and then tested them accordingly. The dry abrasion test was set up as is shown here. The sheets placed on a horizontal vibration plate. Salt is used as the abrasive material, and then they're vibrated for different amounts of time. The resulting debris is then suspended in water. For the hot and cold migration tests, wet tests, paper sheets were cut up into samples about 1 by 1 centimeters, immersed in water, and then left either at ambient temperature or held at 80 degrees C. And the resulting supernatants were then sampled, and we were left with these waters containing migrated material. And the idea was then to use SEM to identify the nano parts and quantitatively compare them. So to make the analysis comparative, we maintain the relative differences in concentration of these suspensions when diluting to a suitable solids content for SEM preparation. And then when preparing the samples for SEM, we took those suspensions and we dispersed an equal amount of each sample across a nuclear pore filter, which was then placed on the SEM stub, which is then dried and coated. And you can see on the right here, that's the residue left following the filtration process, and it gives a good dispersion of the material. So then we had to figure out how to image them using SEM so that our results would be comparative. And so the method that we came up with was to bring each specimen into focus at a low magnification so that we could see the whole 5 millimeter residue area. And then after decreeing a suitable starting point, an area was mapped out to give a total of 3 millimeters left to right and 3 millimeters top to bottom, without leaving the edge of the residue. The extremes were then noted in the instrument's X and Y coordinates, and were then divided to give values to enable 8 evenly spaced image frames left to right and a total of 10 top to bottom. So with the starting position decided, the top left, the first image could then be collected. And these were all collected at a single magnification because we were only interested in comparing the amount of nanomaterial migrated. We used a single magnification that gave us the best balance between resolution and area. And so this was all done at times 70,000. And these were then collected using SEM. So with the first image saved, you then simply move the instrument along to the next coordinates. And the process of capturing images was repeated for all subsequent grid positions. Due to the time taken to acquire image, if it was clear to the operator that there was no material present in the field of view, then no image was taken, and a note was simply made that no material was present, and so no image was taken at that position. So the SEM images are collected, as I said, at a single magnification. This approach was chosen again because of the ratio that it gave of resolution over area. And I must apologize there. I wanted to show an image with our marked labels on it, but unfortunately, when we do the labeling, we do zoom in, and you make the label, and the labels come out quite thin. And then once you zoom out again, you can't really see them very well. And these are the results that we had. And they came out really well. We're very pleased with the method and the patterns that we expected to see. We did. Because of the high binding capacity of MFC, we expected the sheets containing MFC to have lower migration counts. And that was indeed the case for the cold migration, the hot migration, and the abrasion tests. And the other thing that we noted was that the other thing that we expected and then saw in the results was that the hot migration would have higher counts to their equivalent cold migration samples. And this too was the case. With the abrasion samples migrating very little compared to the wet test samples. And the column on the right shows the normalized area of each size distribution curve, just to highlight the difference between the no MFC sheets and the Fiberlean sheets. And so these are the cumulative counts curves. The solid line here is the no MFC line, and ideally all of our samples would sit to the right of that. You can see that for a couple of the samples, we do have slightly higher migration of finer material. And that was the same with the hot migration too, slightly higher. But overall, the counts of nano-sized materials came out lower. And then these were particularly low for the abrasion samples. So we developed a method for comparative analysis of migrated nano contents using SEM. And we saw that fewer nanomaterials migrated from sheets made with MFC than sheets made with the standard furnish. And listed here are the percentages of material that migrated less. And this is just to say that, yes, we are part of the consortium that Joanne spoke about, and we're very grateful to them. And this is some of the work that has come about because of that. And we're grateful for all that it's doing for us in terms of allowing us to enter to markets that we otherwise couldn't without the relevant regulatory clearance. So thank you to Vireo advisors and the NIA for all their support, expertise, and work to help us obtain the clearances that we have. And I'd also like to thank Imaris, Somya, and Fraunhofer for their support of equipment and expertise too. And thank you for your attention.
Daniel, indeed, thank you ever so much. That was a very thorough and very interesting presentation. And now we shall move on, and something that I possibly should have done at the beginning, but we'll sort of make up for now, as we take an opportunity just for a couple of minutes, just so I can sort of tell you all about the NIA, because I know we have some of our members here, but obviously we have some people who aren't members as well. So I'd just like to take an opportunity for 2 minutes just to tell you a little bit about the NIA and the sort of work that we do. So hopefully everyone can see my screen now. Nicoline, if you can just check that that's working. Yeah, perfect. Then I can see Joanne nodding, so that's good. So hello, good afternoon everyone. My name's Sean Kelly. I work at the Nanotechnology Industries Association, and very delighted to sort of say hello to everybody here and welcome you to this webinar today. So what does the NIA do? Essentially, we're all about supporting nanomaterials commercialization. We want to help companies get better regulatory pathways through to market, make sure that we have good confidence in nanomaterials across the whole of the value chain, so that not only are nanomaterial producers confident, but also potential users of nanomaterials can be confident about using those materials in their products. We help bring together the right people, the right sectors, and also find the right spaces to be able to enable people to better collaborate. And we also help people understand where they are in the entire value chain. And especially now that product stewardship across the whole of the value chain is becoming more and more important, not just in Europe, but also more widely as well. So what do? We try and act as the voice of the nanotechnology industries. We help support the development of nanotechnology innovations. We help trying to develop not just help companies comply with current regulatory frameworks, but also try and help develop better and more robust regulatory frameworks through using our members to also speak to the regulators and make sure that regulations coming out are workable for industry. We help with a lot of business and scientific networking, so bringing together our members to collaborate in places and allow them to actually advance the work that they're trying to do as individuals. And we help try and build the global commercial nanotechnology ecosystem. And so it's really important to try and help bridge those gaps in between academia and the wider world. Who are our members? Well, the members are a vast range. We have a lot of commercial producers of nanomaterials and indeed users of nanomaterials who are members. We also have some support organisations who are both providing professional service support, often to industry, but also often to academia as well. And that can be for regulatory compliance as well as actual more research support. We also have a range of research and other national and international associations as well who are members. A lot of people join us because of the regulatory and policy priorities that we help with. So we're trying to develop a global cross-sector regulatory support for our members, and we help with providing access to current regulations, advice and support with upcoming regulations, and also acting as a sector stakeholder, representing our members back to some of those regulators as well. And around policy, we work with a wide variety of policymakers to try and ensure that the right government policies are in place to enable nanotechnology to progress to the market. We produce a range of position papers. So one of our most recent ones has been on nanoplastics, and we engage our members in a lot of member consultations to ensure that we truly are representing our members' interests. For the business and scientific networking, we run obviously the webinar series, both this one, which is Nano in Business, but also the Nano in Action series as well. We have an annual symposium, and we're building up a good social media presence as well, giving access to a lot of people, not just from within the NIA family, but also a lot of people who are interested in nano who are outside of the family. We get involved in a lot of the collaborative projects, and especially around innovation and the safety, regulatory and standard support. So the invitation is there for people to get involved. You can both sign up for our newsletter, sign up on LinkedIn, follow our Twitter account, but indeed what we try and encourage people to do is to think about membership itself. So trying to become a member to help drive the activities of the NIA and also make sure that your voice is there and represented. Membership is open throughout the year, but obviously now's a good time to join to get the full opportunity of a full year in 2021. And so if anybody's interested in membership, then just send us a message and we can arrange to speak with you and your colleagues. So thank you ever so much for that, and we shall move on to Dimitar Prodanov now from IMEC, who's going to give us another presentation, again looking at the public-private partnerships, but again probably from a slightly more different angle and certainly from a different technology space. So I'll just quickly switch slides because Dimitar is working from his phone. So I'll just make sure that I've got his slides on screen for him. So hopefully, Dimitar, you can see your slides there.
Yes, correct. Thank you very much, first of all, for inviting me. And I'm sorry that I have these difficulties, but they are caused by our ICT team policy that we're not allowed to use Zoom on corporate machine. So I'm going to talk today about the open innovation model at IMEC and also some of our experience in publicly funded projects, such as NanoStream, which I coordinated. Can I have the next slide, please? Yes, so first of all, who am I? I'm a nanosafety, I'm a biosafety professional. I was coordinating NanoStream from 2016 till 2019 when the project was finished. And I joined IMEC 2008 as a scientist and switched roles more towards administration and safety in 2013. Can I have the next slide, please? Yes. So IMEC is one of the leading independent research institutions worldwide. We have our core of our activities in Belgium, where the headquarters is located, but we also have campuses in the Netherlands, in Eindhoven, in USA, in Orlando, and some offices with more trading functions in Japan, Taiwan, mainland China, India, and San Francisco. Can I have the next one, please? Right. If you look at our Belgian activities, we have the main campus is located in Leuven, but we have research labs in all of the Flemish universities, and also a campus related to solar cell and energy related activities in Hasselt. Can I have the next one, please? So we are pretty multinational institution. We have 74 different nationalities and cultures all over the world. And can I have the next one, please? And we are actually active in quite a lot of technical and scientific domains. From the application domains, we have a strong life science and healthcare division. We do things related to mobility, self-driving cars. We have projects about city automation. We have industrially related projects for automation and different energy related projects. But actually what is the core of our facilities and business is the CMOS. And CMOS stands for complementary metal oxide semiconductor. And this is the focus of the Leuven campus. Can we have the next slide, please? So what you see here in this picture is our 300 millimeter facility in Leuven. So this is how a semi-industrial clean room looks like. And actually all these different devices and tools are used to process silicon wafers to produce different types of chips. So there are a lot of process steps. Can I have the next one, please? So there are a lot of process steps, and I'll come to that in a minute when I start presenting actually the NanoStream project. So how are we funded? A lot of our revenue comes from the industry in terms of the core program and bilateral work. We have a grant coming from the Flemish government. It's a framework agreement which is renewed every 7 years based on different KPIs. And we also have some funding coming from the European Union in terms of the Marie Curie programs, the ERC programs, and dedicated research projects in ICT and NMP. Can I have the next one, please? So I'm going to talk about now the Open Innovation Platform, what it means, and give some examples without giving too much details, of course. Can I have the next one, please? So as I started talking, most of our activities are related to semiconductor processing. And what you see here on the left is how a silicon wafer is processed. So there are a lot of repeatable steps that are necessary in order to deposit different layers with different chemical properties so that we end up with the full chip architecture. Obviously, we need a lot of precision and high yield. So the precision in terms of the smallness of the lithographic detail and the yields are key factors in our R&D because this is actually what makes a profit or loss for a company. Can I have the next one, please? So if you look at our ecosystem, we are a unique place where different types of partners are collaborating in order to develop these types of technological solutions and platforms. So we have suppliers, we have foundries, we have fabless and fablite companies, and we also have a number of application partners. Can I have the next one, please? From the foundries, we have good relationship with all of these companies, but you see the names, these are quite big players like Samsung, Intel, TSMC, Global Foundries, and so on. Can I have the next one, please? And from fabless and FabLite companies, we have different types of R&D going on with all of these companies who are also participating in terms of bilateral research around specific topics. What is interesting to note here is that some companies, such as some SMEs, let's say, they cannot really afford a full fab, or it's not economically viable for them. So they work with institutes such as IMEC to come up with designs, test them in low volume setting, and then go to a dedicated foundry. So this is one of our cooperation models. Can I have the next one, please? I will give some examples now on the more research-oriented activities which are publicly funded. What you see here on the picture comes from an ERC grant that was funded from 2014 till 2019. So that shows a high-level application where actually we designed a platform on chip that is used to sort different types of cells. So the cells are loaded in the silicon chip, and then the chip can recognize the size, basically, of the cell and decide whether to push it to the appropriate channel or not. So that was a technological development in life sciences, which also was used in other projects because that's a still very innovative project. So the cell sorting platform was only one aspect of it. Another was being able to holographically image the cell and reconstruct this image and use it for further applications. Can I have the next one, please? Actually, what you see here is a example from the holographic platform where which was the developing scalpel, where the cells are imaged and the image is reconstructed. And this technology was later on spun off as a company called Maya Diagnostics with the aim of commercializing the blood screening. So can I have the next one, please? Another example is a project with the city of Antwerp, where we built a network connecting different types of public transports, air sensors, and traffic lights sensors, which are used to optimize the transport in the city. So that's an example of a public-private partnership that we have. Can I have the next one, please? So how we see the opportunities in these types of models. If we look at the added value, service-oriented model has relatively low added value because then the partner has the idea, and then we only operate according to a specification. From such model, we try to move into development on demand, where actually we do also some co-development, and also we do tech transfer in terms of different R&D projects that we also try to spin off. Can I have the next one, please? So if we look at our platform type of research, which is exemplified by the core program, there we define goals with different partners where we actually try to achieve in the set time frames. This is supplied by different type of PhD topics and also sometimes from public funded research that de-risks part of the innovation activities. But also from the platform research, from time to time, we develop bilateral projects for around specific applications that actually push towards higher technological readiness levels. And finally, the outcomes of a platform research can be out licensing to a specific partner or group of partners, depending on the licensing agreements, or spinning off the technology in terms of a company that actually develops a product around it or a service around it. Can I have the next one, please? So an example of a funded project where I was quite involved, I was coordinating it, was NanoStream, where we had a number of industrial partners such as Intel, LFoundry, NXP, Soitec, ST Microelectronics, and Texas Instruments, and research institutions. So we wanted to investigate the nanosafety aspects of the semiconductor manufacturing. So we wanted to identify which are the processes at risk and the hotspots of risks where we actually need more detailed risk assessments and risk mitigation. Can I have the next one, please? Why was this important for all of these different companies? That because bringing in new materials and pushing down the limits of the Moore's law requires very concentrated efforts, both in terms of R&D, but also in terms of safety assessment, because here you see that we are using a lot of exotic materials with quite unknown properties, and some of them may come in nanoform, therefore our interest in nanosafety. So we managed to get a competitive grant from the European Union in order to investigate this. Can I have the next one, please? So the project ran from 2016 to 2019, and you can still access the project website where we have published the main results. What you see here was the final project meeting, which was held at the ST Micro facility in Crolles, France. Can I have the next one, please? Okay. That's my ending slide. This is a map of most of our partners at IMEC. So you see that the matrix is quite large and the ecosystem is quite large, and there are different models that can work for different application domains. I mean, the model in imaging can be quite different from the model of life sciences, and can be quite different in the model of the core CMOS. But our institution is I would say, uniquely positioned to work with quite different types of companies and partners. Okay, thank you very much for the attention.
Indeed, and thank you ever so much, Dimitar, for that very interesting presentation. And now I'd just like to thank everyone for coming to the webinar.